Welcome everybody. Today we'll be looking at another topic on our basic microbiology on Bio 002. Our topic for today is genetic materials. In this course, we'll be looking at genetic materials, DNA replication, RNA, and other relevant information associated with genetic materials in microorganisms. So, nucleic acid. What are nucleic acids? These are organic compounds that carry genetic information for the synthesis of protein in the cells. They contain transferable molecules which can be passed from one generation to another through the process of cellular or DNA replication. So, the nucleic acids are informational compounds and is found in every living organism. We have two types of nucleic acids. We have the deoxyribonucleic acid, which we also known as the DNA, and we also have the ribonucleic acid, which is better known as the RNA. Now, the two nucleic acids have composition of structural nucleotide monomers, which are linked together to form a giant biomolecules. This nucleotide contains three major parts, which are nitrogenous base, 5-carbon sugar, and a phosphate group. Nitrogenous base represents proteins, amino acids, that are present in the diet, while the 5-carbon sugar is associated with carbohydrates, and phosphate is a mineral group. So they are joined together by covalent bond to form polynucleotide chains. The covalent bonds made between them and the sugar is called phosphodiester bond. And the phosphodiester linkage from the sugar phosphate backbone. The phosphodiester linkage forms the sugar phosphate backbone of both the DNA and the RNA. What is a gene? A gene is the functional unit of genetic information in all life forms, including microorganisms. A gene is present in all living things, and they are located on the chromosome of every living organism. They are a segment of DNA that contains specific codes for specific proteins that function in every living cell. So every living organism has a gene, not one gene, multiple genes, lots of genes, and they are all in the DNA in form of a segment. The nature of a DNA is such that DNA is the center of genetic information in the nucleic acid. It carries the blueprint for the cell, while the RNA, which is the second nucleic acid, is an intermediary molecule that only carries information from the DNA. It is an intermediary. It converts the information on the DNA into expression in form of protein. The genetic material of a cell or of an organism is found in the nucleus, mitochondria, cytoplasm, and they play fundamental roles in determining the structure and nature of cell substances. And all these are embedded in the DNA. It is the RNA that convert this information into expression that now play roles in cell development. The DNA has three types of chemical components. It has a phosphate, it has a sugar that is called deoxyribose, and a four nitrogenous bases. These bases are adenine, guanine, cytosine, and thymine. Two of these bases, adenine and guanine, they have a double ring structure characteristics of a type of chemical called a purine, while the other two bases, cytosine and thymine, have a single ring structure and they are called pyrimidine. Each nucleotide is preferably called by the first letter of the name of its base. So adenine is represented with A, guanine represented with G, cytosine represented with C, and thymine with T. So in contrast to other molecules such as polysaccharides and lipid, nucleic acids are informational. Nucleic acids are informational and they are in form of macromolecules which has protein as its monomers. So these protein monomers are called nucleotides. Consequently, it means that if the protein monomers are called nucleotides, DNA and RNA are polynucleotides. So, like we said, a nucleotide consists of a pentose sugar, a nitrogen base, and a molecule of phosphate. What are DNA genetic codes? The term DNA genetic code refers to the set of rules that translate the information encoded in DNA sequence into proteins, which is the building blocks of life. This is how it is. The process of translating DNA into protein is called translation. There are specialized molecules called ribosomes that read the codons in the DNA strand one by one and match them to their corresponding amino acid. This assembly lines process build the protein molecules according to the instruction encoded in the DNA. Now, this information encoded in DNA are called DNA genetic codes, and there are millions of it. And this is what is converted in the ribosome to form the protein that is expressed. Here is an example of DNA genetic code. We have the amino codon, we have the UUU, which is phenylalanine, we have the UCU, serine, GUU, valine, and so on and so forth. So in the building blocks of DNA is composed of two side-by-side side chains. That is, it is double strands. And in the double strands, it has a shape of helix, whereby the base are pairing with each other and they are complementary to one another. That is, guanine complementary with cytosine and they bind together, and adenine binds with thymine. So G plus C and A plus T. So that is, whenever a G is found in one strand, a C is found in the other strand of the double helix. And whenever a T is present in one strand, it is complementary strand and has an A on the other end. The nitrogen base are attached to the pentose sugar by a glycosidic linkage between a carbon atom and a nitrogen base. So the DNA is a double-stranded in the genome of a cell. A genome is an organism's complete set of DNA, including all of its genes. So each strand of DNA contains hundreds of thousands, several millions of nucleotides linked together by phosphodiester bonds that I explained in the beginning. That is the bond between two sugar group and a phosphate group. So the sugar phosphate backbone remains the strong connection for DNA and RNA. This is an example of the pairing of the complementary bases. RNA. What are types of RNA? RNA are single-stranded molecules which does not have its own gene, but is an intermediate and assist in converting the genes in the DNA for expression. When genes are expressed in an organism, the information is usually stored in DNA and is transferred to the ribonucleic acid. So in this form, we have three types of RNA, which is the messenger RNA, transfer RNA, and ribosomal RNA. Let's look at this RNA one by one. So messenger RNAs are single-stranded RNA molecules that carry the genetic code from DNA in the nucleus to the ribosome in the cytoplasm for protein synthesis. While the transfer RNA is a molecule that acts as an adapter molecule during the protein synthesis, bind to specific amino acids and deliver the correct amino acid to the ribosome during protein translation. It helps to convert the information that is coded on the DNA code for proper synthesis. So it converts the genetic information that the messenger RNA has delivered to the ribosome for further processing, while the ribosomal RNA is the catalytic process that synthesizes the protein that is coded from the DNA. So this converted DNA sequence is now being synthesized for the specific protein that is being coded for. So the three RNA types work together. Messenger RNA carries the genetic information from DNA to ribosomes, while the transfer RNA converts it to the normal protein language, while the ribosomal RNA does the synthesis of the protein. Based on the function of RNA, there are differences between RNA and DNA. So although they are both informational, but they are different in terms of activity and nature. RNA contains the sugar, which is the ribose, while the DNA contains the deoxyribose sugar. RNA contains the base uracil, while in DNA it has thymine. And the DNA is a double-stranded molecule, while RNA is single-stranded. Other information is that there are forms and differences. DNA actually carries several lots of genes, while RNA is just an intermediary. And DNA is also stable because of the fact that it is double-stranded, RNA is relatively unstable and can easily be degraded after its activity has been completed. DNA is stable, RNA is not stable. So the DNA replication itself. Now we are going to look at the DNA replication, and this animation is showing us how DNA replication progresses. The process of DNA replication starts with the unwinding of the double strands. The strands of the DNA that form at replication forks are usually complementary. Now, the first step in DNA replication is the separation of the strands into two separate single strands, and this normally happens at a point called the replication fork or the replisomes by an enzyme called DNA helicase. The DNA helicase helps in unwinding, that is, it separates the DNA from each other. It helps to separate the strands at the beginning of the replication process. And the DNA replication has three stages, three steps. We have the initiation, elongation, and termination steps. So let's look. This unwinding is called initiation step. That is the first step in DNA replication. And this involved uncoiling and unzipping of the DNA double strand. It begins by gradual separation of the strands into single strands, and both strands are now separated. In the picture here, you can see the helicase in the black, and the helicase, this is the helicase, and it begins to unwind in this direction, like that. These are the separated strands, and these are the complementary bases together. Now, after the unwinding, then comes the elongation step. The elongation step is the synthesis of complementary base continuously. So it is ensured by an enzyme called RNA primase. The RNA primase continues to synthesize the base in a complementary fashion, and this continues in the direction of 3 to 5. Now, there is two strands. One is a leading and the other one is a lagging strand. The leading strand is the continuous strand, which is going in the direction of 3 to 5. There is a lagging strand. The lagging strand is called Okazaki strand. I will explain that. So the RNA primase helps to copy and synthesize by using DNA template and synthesize the complementary base to form a new strand. You can see the direction it's going. This is the RNA primer. This is the template synthesizing new complementary base. Once this is synthesized, so we are going to have a new strand, just like what we have here. This is the strands. A new one will be synthesized. This is the unwinding. This place has been unwound, it's been already separated, and new strands are being synthesized in the complementary. C and G are complementary, A and T are complementary. So they are being added to each other like that continuously. So as this unwinds, it continuously synthesizes. The enzyme called topoisomerase prevents the DNA from coiling. Because DNA is supercoiled, the enzyme prevents it from returning back because it is helix. So it prevents it from coming back together so that there will be no accidental addition to another strand or damage to the DNA during the process. So the topoisomerase enzyme prevents double helix DNA from getting too tightly wound as the DNA is opened up. It relaxes the supercoiled DNA as the elongation proceeds. Then the other lagging strands, referred to as Okazaki strands, are the strands that are synthesized in short nucleotides. It's not a continuous synthesis. It is synthesizing the DNA in short bases. 10 bases, stop, continues, 10 bases, stop, continue, or 5 bases, stop, and continue. So by that, it forms a fragment of Okazaki. And at the end of the day, one strand is continuously while the other strand is slow. That's why you call it a lagging strand, because it is not continuous. So the elongation steps ensures that complementary bases are added to each other. The lagging strand unwinds in a small section that DNA polymerase replicate on the leading direction, and it continues until all the bases has been added. After addition of all the bases, then it will now get to now at the termination step, the sequence is altered. Every sequence has been added, all the complementary bases have been added. Then it gets to the termination steps where the stop codon initiates the stop action. The DNA polymerase reads the fragments and removes the RNA primers that is ensuring the synthesis of the bases. And for all the lagging strands, the gaps are closed with the action of DNA polymerase. And the DNA ligase begins to add them together and joins everything together, and they form a straight sequence. This is the leading strand. This Okazaki fragment. You can see that this point has nothing. This point has nothing. This is Okazaki fragment. So this this is now joined together completely. This is a leading strand, this is a lagging strand. They are going in different directions. So after this comes the termination of the sequence. The termination steps is the last of the DNA replication where the process is terminated. This process happens when the DNA polymerase reach to an end of the strands, and at this moment the primase, which is the primer, must be removed from the lagging strand, and also the nuclease, which is an enzyme, do what is called a proofreading. The proofreading means it begins to check for errors and remove any mispaired bases. The last portion of the lagging strand is the telomere section. It contains a repeating non-coding sequence of bases, and every sequence, especially the primer sequence, is accounted for. The primer sequence is removed, the non-coding is not relevant, and the nuclease begins to do what is called error checking. And once everything is done, then the DNA is completed, and the double-stranded DNA now becomes a duplicated exact copy of the original. And that is the basic description of DNA replication in microorganisms. Thank you for listening. We will do more on other topics. Thank you for your time.